Hello, and welcome to this second podcast on Mendelian inheritance. In the last podcast, we introduced inheritance. We talked about Mendel and why he chose the pea plant for his studies. In this podcast, what I'd like to do is to first talk about Mendel's pea crosses that led to his first law, the law of segregation, and then talk about his pea crosses that led to his second law, the law of independent assortment. And then we'll have one final podcast, the third in this series, which will be a little shorter, and that'll just be on pedigrees and how to solve pedigrees. Okay, let's go ahead and get started. So we've introduced Mendel, we introduced the peas that he used. So now let's start looking at some of his crosses and try to understand his observations and how those observations led to his laws. So Mendel initially did what we would call monohybrid crosses. Monohybrid, mono means one, so this was just one trait he was looking at least initially. Later on, he did more than one trait. So let's just look at the shape of the pea seed here. And he knew from his observations that, roughly speaking, there were two kinds of pea-shaped seeds. One was smooth and one was wrinkled. He knew that if you took a round-seeded plant and crossed it with a round-seeded plant, or you took a wrinkle-seeded plant and you crossed it with another wrinkle-seeded plant, and you did this for many generations, that it would result in all the plants being smooth or all the plants being wrinkled. We call this a pure breeding strain. Today, we would call it a homozygous strain. We'll talk about that term later. Now, what he did next was he took this smooth seeded plant and he crossed it with a rough or wrinkled seeded plant. Now, before you do this experiment initially, you might think there are one of several different possible results. Maybe they are going to be all smooth. Maybe they'll be all wrinkled. The progeny, that is. Maybe you'll see both. Maybe some of the seeds will be smooth and some of the seeds will be wrinkled. Maybe you'll see a combination where the peas are somewhere in between. They're not perfectly smooth, but they're not very wrinkled either. When he did this cross, what he found was that they were all smooth. All of them. Now, it's important to note that at this time, Mendel did not know about genes or chromosomes. But we can go back and look at this, and we know that these are all caused by a single gene. And so let's do this. This first one, let's call it big R, big R, big R, big R, little r, and little r, little r. Same thing with this one, big R, little r, little r. And so let's talk about this term over here I said I would mention again. We know that when something is big R, we call that homozygous dominant. Something that is little r, we would call homozygous recessive. I'll talk about this in a moment, but we know that all of these progeny are big R, little r. And again, I'll explain that in a moment. Big R, little r, we call heterozygous. Again, this is homozygous dominant because both of the alleles are big R. This is homozygous recessive because both of them are little r. This is heterozygous because it has one of each allele. One big R, one little r. We know that the big R is dominant because when we cross them together and we have a plant that has one big R, one little r, we see the phenotype of the big R, round. So we know that round, big R, is dominant to wrinkled little r. Let's also take this opportunity to review a couple other terms. In this case, big R, little r, and big R, little r, these are genotypes. We talked about that before, but it's good to mention it again. The trait of being round or wrinkled is the phenotype. All right, so let's look at this cross in a little bit more detail. So remember, he took a round plant and he crossed it with a wrinkle-seeded plant. We call this generation the P generation for parental. And when he crossed these, remember, all of the progeny had round seeds. All round. We call this the F1 generation, and that stands for filial first generation. So the next thing he did was he took two round seeded plants and he crossed them together. And what he got was a mixture of round seeded plants and wrinkle seeded plants. And we call this the F2 generation, or filial second generation. I suspect that farmers had seen this trend before when they crossed plants, and they probably didn't think too much of it. But Mendels' love of numbers forced him to count all of these. So I imagine his fellow monk brothers thought he was crazy sitting there counting all of these pea seeds. But what he discovered when he crossed them was that approximately 75% of them were round and 25% of them were wrinkled. Now, he didn't just do this once. If he just did this once and that's what he got, that wouldn't be such a big deal. But Mendel was persistent. He did these crosses 60 times, over 60 times. So he was pretty confident that these numbers were true. Now, this led to his first observation, and that is that the F1 plants, these here, must inherit both genetic factors. We know there are genes on chromosomes today, but Mendel didn't know that. So both genetic factors for round and wrinkle from the parents. Even though we don't see the phenotype of wrinkled here, he reasoned that they must have inherited something from this parent and this parent. And the reason he made that prediction is because the wrinkled phenotype shows up here again. Where else would it have come from? So it was a pretty logical prediction. He also reasoned that each plant must have two copies of each genetic component. Again, we know that today to be the gene. Because he reasoned that if you only had one copy of it, there'd be no way to kind of hide this wrinkled gene in this F1 generation. So again, let's go and put our genes in here that we now know today. And so remember here in the F2, because they have two different copies, it can now inherit one of these little r's from each parent to provide the little r. Over here, and we'll explain why we know this on the next whiteboard, we know that these plants are either big R or big R little r, and we'll come back to that in a moment. It was pretty bold of him to make this prediction without having the molecular tools that we have today. He was absolutely correct, and he did this by just looking at the math behind it. He also reasoned, I'm not going to write it down, I'm just going to state it here. He also reasoned that these two copies that each plant has, these two, that during the process of making gametes, they must separate from each other and with equal probability end up in a gamete. So just is likely to have a big R gamete versus a little r gamete. And he made that prediction because of this very robust outcome. And when we show you a Punnett square, that'll make more sense. Now this next observation I'm going to write down here is the concept of dominance. He reasoned that these plants here in the F1 generation must be big R, little r. They have the gene that could give a round shape, and they also have the genetic allele that could give you the wrinkled shape. However, when they're together, you only get the round, and the little r is masked. It's hidden, so to speak. Its phenotype is hidden. And the only way that could be explained, Mendel believed, was that R, the R allele, must exert some kind of dominance. So must be dominant to little r. And of course, we now know that this is absolutely correct. That when you have two alleles like that, often one of these alleles will be dominant over the other. Now, there are some exceptions that we'll talk about in a couple podcasts from now. But in general, this holds true. And so because of that, a big R or a big R little r will have round seeds. And the only way you can see a little r, or the only way you can see a wrinkled seed, is if the two alleles are little r and little r. These observations that he made led to his first law, and that is the law of segregation. Alleles of a gene segregate from each other during gamete production. And each gamete has equal probability of getting one or the other allele. It's important to remember that with the law of segregation, we're only talking about one set of alleles. When we talk about his second law, the law of independent assortment, that's going to be a law where we're talking about two sets of alleles, that is, two different genes. Now, I don't want to belabor this point too much, but I think it's important for us to recognize that at the time of Mendel's studies, he did not know the full understanding of what the chromosome was. It had been discovered in a cell, but nobody knew really what it was or what it was made of, and certainly nobody appreciated it that it was the molecule of inheritance. So he made his models and his predictions based off just observations and using his brain. But we now know that the gene, and we can say this is the gene for the pea seed color. And so let's just say this was big R and this is little r. And so we know when the gamete is made, and we know today, and from our review of meiosis a few podcasts back, that as gametes are being made, that these chromosomes separate from each other. And you'll end up with gametes, and I'm leaving out a lot of steps here, of course, and I'm going to shrink down this chromosome here. We know that each gamete will get one or the other chromosomes. So one of these gametes, in this case, big R, and this one, little r, each gamete will get only one of those because they separate from each other, just like Mendel predicted. All right, now let's move on to Punnett squares. I suspect some of you, maybe many of you, have worked with Punnett squares in the past. But let's review them. And we're going to review them using Mendel's F1 cross from a few whiteboards ago. And what I'd like to do, instead of just writing big R, little r here, what I'd like to do is write this slightly differently. I'd like to draw the chromosome. So we don't forget that the unit of heredity is not just a letter, it's the whole chromosome. So when we say big R moves into a gamete, we're saying big R and all the genes on it. We're only focusing on this one though. So remember that the parent that made these two gametes was big R, little r. It went through meiosis to produce a gamete that had big R and an equal proportion of gametes with little r. This parent up here also was big R, little r that made these two gametes in equal probabilities. When the gametes come together, we can circle these so they look like gametes if we use our imagination. Now when they come together to form the embryo, in this case, let me put it in a different color and a different shape here, it's going to get the two chromosomes. It's going to get one from one parent and one from the other parent. So let's go ahead and draw that in each of these first. Okay, so this one's going to get a big R and a big R from these two parents here. This one's going to get a little r from this parent, but it's going to get a big R from this parental gamete. This one here will get a big R from this gamete, and it'll get a little r from this gamete. This progeny here will get a little r from each of its parents. So let's look at how we would write these probabilities out. The genotypic probability probability would be 3 round to 1 wrinkled pea seed shape. 3 round because big R and big R little r would have the phenotype of a round pea seed, and little r would have the phenotype of a wrinkled pea seed. We will use these Punnett squares, or you can use these Punnett squares as you work on various problems. So for instance, on an exam, I might give you a cross and ask you to tell me what the phenotypic probability is, or I might ask you to tell me the genotypic probability. Okay, now with a firm understanding of Mendels' first law, the law that stated that a single gene has 2 alleles, and those 2 alleles separate from each other during gamete production and separate into gametes with equal probabilities. So now let's go on to Mendels' second law, his law of independent assortment. And to talk about this, what I want to look at are some chromosomes. And remember, when we talked about the first law, we said that the gene for seed shape was found on a chromosome. And if it was a heterozygote, we'd have a big R on one of the chromosomes, and on the other one, it would be little r. And remember, this would make the plant, the phenotype of the plant, round because big R is dominant to little r. However, we know that most organisms have thousands of genes. So what about another pea plant gene? And so Mendel looked at this other trait, and that is seed color. Big Y is the dominant trait, and that is for the color yellow. And then the recessive trait is little y. And I'm going to put a line under little y so we can distinguish it from big Y. We know that as this plant cell, remember the plant cell itself would have, or the nucleus anyhow, would have both of these chromosomes in it plus all the other chromosomes. We're just looking at these 2. We know as it goes through meiosis that each of these will get copied, and that the end product of meiosis are 4 gametes. We know that, as the first law says, these alleles, after they're copied, will segregate from each other in equal probability. So we know that this gamete will have big R, and we know that with equal probability, another gamete will have little r. And again, as I said, and if you review meiosis, we know before it enters meiosis, it goes through S phase and it copies these. So we're going to have 2 of each, big R and little r. This follows the first law. Now, let's think about this other chromosome. We know that this other chromosome also has to follow Mendels' first law. So we know as it segregates these 2 alleles from each other with equal probability that some of the gametes will get big Y and some will get little y. However, we know that as they go into the gametes, that that the chromosome with the big Y gene on it is just as likely to segregate with big R as it is with little r. So this is also possible, a gamete with little r and big Y. Same thing with the chromosome that has the little y gene on it. I drew it here with the little r gene, but it's just as likely to have segregated with the chromosome that has big R gene. So we're going to put little y gene here. Each of these gametes are equally probable because each of these 2 chromosomes follow the first law of Mendel. His second law states that genes at one locus, and let me define locus up here because I think that's the first time I've used this term. This is the physical location of a gene on a chromosome. So, for instance, this physical location, that's the locus for the R gene. So, genes at one locus do not interfere with the segregation of genes at a second locus. Genes at the locus for R do not interfere with the segregation of genes at the second locus of Y. So let's add to this and say the 2 sets of genes independently assort. Let me say that using this picture here. The genes at this locus here assort into gametes independent of genes at a second locus. So remember with Mendel's first law, we took the gametes and we built a Punnett square. Let's at least start that with these as the gamete. So let's go ahead and put our gametes up here. And I'm not going to draw the chromosomes in here just because of space, but I'm just going to write the genes. So big R, little r, little r, big R, little r, and then the genes at the Y locus. Big Y, little y, little y. And then we'll do the same here. And then, if you'd like, you can complete this Punnett square. So we'll combine R and R. So this will be big R, big Y, big Y. And I'm just going to complete this and then fast forward on the video because it's going to take a little bit of time. So what you might want to do on your own is just hit pause and hit play again, and then you can check your answer here. Okay, welcome back. As you can see, we have quite a variety of different genotypes. However, when we look at this, there's only going to be 4 possible phenotypes. What I'm going to do now is circle all of the ones that are dominant for both seed shape and seed color. All that means is they have to have one big R and one big Y. So all the ones I'm circling will be round and yellow. And of these 16 squares, I should circle 9 of them. So those are the 9 that would be fully dominant. So that's going to be 9. Let's just write that there. What I'd like to do next is to make a little asterisk by the ones that are round, dominant round, but green recessive for color. There should be 3 of these. So that means they have to have big R and then 2 little y's. So that's going to be 3. And then I'm going to put a green asterisk by the ones that are wrinkled but yellow. So little r, and 1 big Y. Again, there should be 3 of these. So let's put 3 here. And then there will be 1 of these 16 that is fully recessive. And that is this one here. Now the reason I knew it was going to be 9 to 3 to 3 to 1 is because anytime we do a dihybrid cross, that is a cross looking at 2 different traits at the same time, that we're always going to see a 9 to 3 to 3 to 1 ratio. 9 fully dominant for both traits, 1 fully recessive for both traits, and then 3 of each where one of the traits is dominant, the other is recessive, and the other 3 it's reversed. This would be a good ratio to memorize. Now, on an exam or an activity, I'm never going to ask you to draw this out. It's good to be able to do so you can see how the genes interact to produce this 9 to 3 to 3 to 1 ratio. But on an exam, it's going to take you too long, and there's a lot of opportunities to make a mistake. So I'm going to show you a shortcut. And this shortcut, I'll write it up here, it's called the fork branch method. Now, so let's go ahead and draw our strains. We had an R, little r, big R, big Y, little y. And remember, we crossed it to the same genotype. So big R, big Y, little y. Now that you've seen the large Punnett square and you understand the second law, as well as the first law, we can take this shortcut. And we can take this shortcut because what we're going to do is we're going to do essentially 2 monohybrid crosses. And if we want to, we can just draw it right over here until we get more familiar with them. No shame in ever doing a Punnett square. We do them around the dinner table all the time. So let's ignore the Y genes for right now. Big R, little r. So we know, and we're only going to look at the phenotype here. We're not going to look at the genotypes. We know that 3/4 of them will have the R phenotype. This one, this one, this one. So that's 3/4. We also know that 1/4 of them will have the recessive phenotype. So we write 1/4 there. Now let's consider the Y monohybrid cross here. So let's go ahead and draw the Punnett square for Y. We know that 3/4 of them, this one, and this one, will have the phenotype of big Y. So we're going to draw 3/4 here. We know as these alleles segregate, they're just as likely to combine with the larger R alleles or the little r alleles. So we're going to put 3/4 here. We also know that 1/4 will be the recessive of green. So we're going to put that here, and then we're going to put it down here for the same reason, that because of the law of independent assortment, they can segregate with either big R or little r. And now what we're going to do is multiply these through together. 3 times 3 is 9. 4 times 4 is 16. 3 times 1 is 3. 4 times 4 is 16. 1 times 3 is 3. 4 times 4 is 16. 1 times 1 is 1. 4 times 4 is 16. 9/16 will be, I should have drawn this as big Y, and this is little y, little y. So 9/16 here will be round yellow. 3/16 will be round and green. 3/16 will be wrinkled and yellow. 1/16 will be round and yellow. Okay, I'm going to show you an example of something you could see on an activity or an exam. Since we know how to use this fork branch method on a dihybrid cross, could we use it on something larger? Well, I'm glad you asked, because yes, of course you can. And to make it simpler, what I'm going to do is just use letters like this. Let's say we have a genotype like this. Now, I'm not going to ask you to take this cross and tell me the phenotypic ratios of that, but I might ask you something like this. What is the probability that this cross between this organism and this organism, that you would have a phenotype of big A, little b, big C, little d? Now, the reason I put a dash by the A and C just is to remind you that if it has the phenotype of big A, it could either be big A, or big A, little a. It doesn't really matter what's at the second allele. The way we're going to approach this is as 4 independent monohybrid crosses. Okay, so let's go ahead and start this by doing the first monohybrid cross with the A alleles. We want to know what is the probability that the phenotype will be big A. So let's do a small Punnett square here. Never a shame in doing Punnett squares. They're fun for the whole family. So big A, little a, little a, little a, big A, little a, little a, little a. So the probability of being big A phenotype by crossing these two together are these two here. So that's 1/2. Let's look at B now. Big B, little b here. Big B, little b there. So the probability of being little b is 1/4. Let's look at C. We have little c, big C, little c. So we have a big C, little c, and little c, little c's. The probability of being big C phenotype would be these two here. These two would be recessive, these two would be wild type. And so again, we have a 1/2. Now let's do our last one. Big D, little d, big D, little d. Complete the Punnett square. The probability of being little d is 1/4. So the overall probability that they will have a child that is big A, little b phenotype, big C phenotype, little d phenotype is the product of these 4 fractions. So let's go ahead and multiply these through, and that will be our answer. 1 times 1 times 1 times 1 is 1. 2 times 4 is 8. 8 times 2 is 16. 16 times 4 is 64. So the overall probability that they will have this phenotype is 1/64. I guarantee you there will be a question on the exam about this. It may not be presented just this way. It may be presented to you in the manner that I plan on asking for one of the application questions. So you'll have a couple more opportunities to practice this. Well, that ends this podcast. In this podcast, we explained both Mendels' first and second laws and described how we could use those laws to make predictions about crosses. Now, in our next podcast, the last podcast for inheritance, for Mendelian inheritance anyhow, will be how we can use pedigrees to make similar predictions. If you have any questions, please let me know. If not, I'll see you on the next podcast.